Hi, and welcome to part 1 of the EDGE tutorial series. In this video, I'm going to introduce the EDGE Bioinformatics platform and give an idea about the workflows EDGE has to offer its users. I will also briefly go over the recommended computational environment and note resources necessary for advanced users that wish to install EDGE locally. We've made parts 2 and 3 of this series for a detailed walkthrough of job submission and data analysis. I encourage you to check those out after you watch this video. So what is EDGE? EDGE is a web-based bioinformatics platform. It stands for Empowering the Development of Genomics Expertise. EDGE was developed to help biologists with little to no technical bioinformatics experience process their sequencing data. This pipeline has been optimized to run many of the standard analyses required to characterize microbial and metagenomic samples. I'll make a quick note here that eukaryotic genome assembly is not fully supported by EDGE at this time. Once installed, EDGE is accessed through a web-based interface, which we're going to take a look at in parts 2 and 3 of this series. To note, even though EDGE is accessed through a web browser, when installed locally, EDGE is standalone and does not require the internet. At the interface, independent users can upload data and apply desired workflows to analyze each sample. All of the tools integrated in the EDGE workflow are publicly available and allow reliable NGS analysis. We will go over those in more detail later. Listed here are a few key resources, including demo versions of EDGE that can be used by anyone with access to the internet. This tutorial will utilize the Naval Medical Research Center's outward-facing version. You can access the same display and follow along in part 2 by typing the following address into any web browser. A written version of this tutorial, along with answers to frequently asked questions, can be found at our Read the Docs website. We also maintain an active Google group to address any questions users pose in real time. For users interested in downloading and installing EDGE, the source code can be obtained from the Bitbucket or GitHub links listed here. Finally, a technical description of tools and use cases has been published in the Journal of Nucleic Acids Research. You can find that by searching the publication DOI listed at the bottom of this page. Part 2 of this series will go into detail for each module, but to get a general idea, here's an overview of the bioinformatics capabilities in EDGE. EDGE has been designed to be as simple as possible for the user. The only required information to run EDGE is a project name and data. A user can upload FASTQ files to EDGE or use publicly available data from the SRA database. When installed locally, an EDGE server can be configured to pull data directly from an Illumina machine for easy and immediate access. The EDGE workflow begins with one or more FASTQ files from a single sample. As a side note, EDGE parameters have been optimized for data coming off of an Illumina machine. If you have data from other sequencers, feel free to contact our team to see how our pipeline can be customized for you. Using easy on and off toggle switches in the EDGE interface, users choose which bioinformatics modules are applied to each sample. The workflow is overviewed here. For example, turning the preprocessing module on will initiate sample quality and filtering. Assembly will stitch the genome back together, and annotation will help identify genes. Users looking for antibiotic resistance or virulence factors may be interested in the specialty gene module. In many cases, uploading data and submitting a job using default settings and parameters provides a useful first glance into sample quality and composition. Part 2 of this series details these workflows to allow users with little bioinformatics background to gain insight for when to apply each tool and how to interpret the results. The EDGE development team has taken great care to engineer modules for reproducibility and reliability using publicly available tools. We encourage users to check out the third-party tools section in the Read the Docs resource to gain knowledge of how the tools and algorithms function. Because EDGE is a publicly available tool, the source code is available at our GitHub and Bitbucket sites. If you're interested in setting up a local instance of EDGE, this slide gives you the information you need to get started. EDGE was designed to be implemented within any institute that produces or hosts sequencing data. Listed here is a guide for recommended system hardware and software requirements. Due to the involvement of several high memory and high CPU consuming steps, we recommend a bare minimum of 16 gigabytes of memory and 8 computing CPUs. The higher computer specs here are strongly recommended. Additionally, the current version of Edge has been extensively tested on Linux servers with Ubuntu and CentOS operating systems. Edge cannot be installed on Windows or Mac OS. We've detailed essential software building packages in the Edge Read the Docs page. You'll need to verify that these are installed first. Then, once your system is properly configured, we provide step-by-step command line instructions to get Edge up and running on your server. All of this information is in the Read the Docs website. If at any point in time you have questions regarding system requirements, installation, the Edge workflow, or even data analysis, do not hesitate to contact the Edge team. We are available through our Google group or by email. Feel free to contact any of the development team members and we'll get back to you as soon as we can. This concludes part 1 of the Edge tutorial series. Don't forget to join us for the interactive demo detailed in part 2 and an analysis of each module's output in part 3.